Assalamu alaikum. I'm Dr. Hasiba Mahmuda, Assistant Professor Current Charge, the Department of Microbiology. Today I'm going to discuss about the introduction of mycology. Let's get started. Mycology. What is mycology? It is a subject that deals with fungus. And fungus is an universal scavenger. When there is a single disease caused by the fungus, it's called the mycosis. And when multiple diseases produced by the fungus are together called the mycosis. It's a plural form. Fungal diseases are called the mycosis, mycosis or mycotic diseases. Characteristic of fungus. The fungal cell is an eukaryotic cell. That means it will have a real nucleus properly formed. The cell wall contains chitin, a polymer of N-acetyl glucosamine, and also it contains the beta-glucan polymer of D-glucose. Cell membrane contains the sterol, which is called the ergosterol and zymosterol. What's the significance of sterol in fungus? No bacterial cell membrane has sterol. The exception is Mycoplasma species. In Mycoplasma species, in the cell membrane, there is presence of sterol. And in human cell membrane, it has also cholesterol. Now, what is the comparison between the fungus and bacteria? In case of fungus, the diameter is larger than the bacteria. The diameter of the fungus is about 4 micrometer, and in case of bacteria, it is 1 micrometer around. The nucleus, the fungal nucleus is eukaryotic, whereas the bacterial cell is prokaryotic. Cytoplasm, in fungal cell, there is a mitochondria and endoplasmic reticulum is present in the cytoplasm, and the mitochondria and endoplasmic reticulum is absent in case of bacteria. In the cell membrane, sterol is present in fungus, which is absent in case of bacteria. Exception is mycoplasma. Cell wall, what's the content of the cell wall? In case of fungus, there is presence of chitin, and in case of bacteria, there is presence of peptidoglycan. Spore, sexual and asexual spore are produced in case of fungus, which is for the reproduction purpose. And in case of bacteria, the endospores are for the survival, not for the reproduction. This is the basic difference between the spore of fungus and bacteria. Then thermal dimorphism. Yes, in case of fungus, there is a term called the thermal dimorphism. We will discuss it later, which is absent in case of bacteria. Metabolism. In case of fungus, it requires the organic carbon. No obligate anaerobic terms are present. In case of bacteria, that many do not require the organic carbon, may obligate anaerobe. Now, what is hypha? Hypha is the elongation of the apical cell that produces a tubular thread-like structure, which is called hypha. They are usually surrounded by a rigid chitin containing cell wall and contain the nuclei, mitochondria, ribosome, Golgi body, and the membrane-bound vesicle within membrane-bound cytoplasm. There are 3 types of hypha. Vegetative hypha, aerial hypha, and another is fertile or reproductive hypha. Hypha may be septate or non-septate. Now, mycelium. What is mycelium? It is the tangled mass of hyphae. It is called mycelium. Fungus that producing mycelia are called the molds or filamentous fungus. What is the difference between bacterial and fungal cell? Fungal spore. In the case of bacterial spore, endospores are formed, and in case of fungus, exospores are formed. And in case of bacteria, the bacterial spore has protective function, and in case of fungus, fungal spore, it is of reproductive purpose. Reproduction of fungus. Reproduced by fungus, the reproduction of fungus is caused by the 2 ways. Sexually and asexually. In case of sexually, sexual spores are meiosis, is the procedure by which cell division occurs, and the examples are ascospore, basidiospore, and zygospore. And asexually, the procedures are budding and fission, cell division procedures. And in case of, what is the examples of asexual spore? Arthrospore, sporangiospore and chlamydospore. Fungus that do not form the sexual spore are termed as fungus imperfecti. Zygospore has single large spores with thick walls. Ascospores are formed in a sac called the ascus, and the basidiospores are formed externally on the tip of a pedestal called the basidium. As the picture showing all three types, zygospore, basidiospore and ascospores. Then asexual spores, arthrospore, which arise by the fragmentation of the ends of hypha. And that is chlamydospore, which are rounded, thick-walled and quite resistant. The terminal chlamydospores of Candida albicans helps in its identification. And blastospore, which are formed by the budding process in which the yeasts reproduce asexually. Some yeast, Candida albicans, can form multiple buds. And sporangiospore, which are formed within a sac, sporangium, or a stalk by molds such as Rhizopas and Mucor. What are the terms used in case of mycology? Arthrospore, which is formed by the separation of segment of hyphae from most dermatophytes. Conidia. Asexual spores are also called conidia. Arise from the sides or ends of specialized structures. Mostly human pathogenic spores are asexual spores. What are the types of conidia? Microconidia, macroconidia. What is the significance of fungal spore? Reproduction, dissemination and identification of fungus. Importance of fungus. Advantage. Act as universal scavenger by consuming dead and decayed organic materials. There is the synthesis of drugs such as penicillin, cephalosporin. Fungus is used in the preparation of beverage and some foods. Protein synthesized like hormones and enzymes by genetic engineering from yeast, fungus, Candida albicans by the process of recombinant insulin. Importance of fungus. Disadvantages. Fungus can produce superficial or systemic disease in both healthy and immunocompromised patients. Can produce disease in animal and plants. Can spoil prepared food. Can damage to cottages and the buildings. And in our next session, we will discuss about the classification of and morphology of fungus. Thank you.